Here now, so welcome everybody. So this is the fourth in our series of the WPC Research Spotlights. So I hope you've listened to the other three. Today's going to be as exciting as those. And I'll even flag up now that the next one is on the 1st of September, when we're going to be talking about topics which actually relate to today, which also relate to the last one. Before we start, I'd just like to thank Supernus Pharmaceuticals, who have sponsored this event. Without the sponsorship, we wouldn't be allowed to do that. We have a chat, obviously, because this is Zoom. So if you have questions, by all means, put them into the chat and I will try and ask them as we go through. The format of this is we're just going to have a little bit of a chat about where everyone's come from in terms of getting to where they are now with this research project around inflammation and Parkinson's disease. And then we're going to ask specific questions about the research and such like. It's very relaxed. Ask any questions you like, and we'll just amble through this over the next hour. So welcome everybody. This is very much a home team, I have to say, since every person on this call actually lives in the same place in the Cambridge in the UK. So we've got Caroline, Nina, Julia and Marta. So I'm going to start with Caroline. I've known Caroline for many years. We probably shouldn't say how many years, but it would be useful, Caroline, just to sort of introduce yourself. You know, what's your position in the university? How did you get into research? A little bit about yourself and how you've ended up where you are today would be a really useful starting point.
Okay, well, I'm a principal research associate in the university and also an honorary consultant neurologist. So I spend most of my time doing research, but I also see patients in our local NHS hospital as well. And of course, my main interest, both clinically and in terms of research, is Parkinson's and specifically this question of the immune system and what contribution that has to play in Parkinson's disease. So in terms of my background, I started out learning medicine here in Cambridge and then did my clinical medical degree in Oxford. And then after a little bit of time doing my junior house jobs, I came and joined you, Roger, as you will remember, as a PhD student back in 2004. And at that stage, my interest was really around sort of the natural history of Parkinson's disease. So we'd set up a cohort study, so a study of a large group of patients with Parkinson's disease. And through my PhD, I was following up that group of people over time, looking at how the disease changed over time, how it evolved, and in particular, how problems with memory and thinking evolved over time. And the main question was trying to sort of better understand the variability in Parkinson's disease over time. And then following that, I went back to do some clinical training for a while to learn to be a clinical neurologist. And then came back to research, I think it was 2013, I then came back as a clinical lecturer. And I sort of shifted my attention a little bit away from the natural history of Parkinson's disease towards trying to understand why the condition was so variable. So we'd sort of...
And that's what the natural history studies had sort of led you to believe. So this was collecting everybody with Parkinson's, follow them over time, and you sort of, you could see that some did rather well, no real problems for many years, whereas others sadly didn't do quite so well. And so you wanted to sort of, I suppose, better understand why that was, and I suppose that's where your project is now. Yeah.
Exactly, yeah. So that's been the question that's always sort of driven my research. I guess it's trying to understand why there is that huge difference, as you say, between people with Parkinson's. So we have some people that we see who have a sort of a condition which is relatively confined to their motor system. So they predominantly have problems with movement, perhaps with tremor, with stiffness, and they respond very well to dopamine replacing therapies. And often those patients do very well for many, many years. We have patients, as you know, coming through our research clinic who've been seeing us for 20 years, and they're still doing pretty well on their treatment. And then unfortunately, we have other people who seem to have a much more complicated form of Parkinson's disease, where they don't just get problems with their movement, but they also get problems with sort of non-motor symptoms. So problems with their mood, problems with memory and thinking, sometimes psychiatric symptoms, problems with hallucinations and that sort of thing. Often those individuals don't respond quite so well to sort of straightforward Parkinson's medication. Often they go downhill sort of quite quickly, unfortunately. And what I've always sort of wanted to understand is why is there that? Why does the same disease behave so differently in different people? So through my PhD, I started to look at sort of various genetic factors that might contribute to that. And we identified 1 or 2 genes that seemed to be important in terms of sort of pushing you one way or the other on that spectrum from sort of benign to a more malignant disease. And we also identified various sort of predictors in terms of clinical things that we could identify when we first met patients at diagnosis that would tell us how likely they were to go on that sort of benign course versus a more malignant course. But none of that was really answering the question of what's the actual underlying biological reason why, you know, patients are progressing at different rates. And so we came up with this theory that the immune system might be important in this. And that's where
did you get any clues from that from your previous, you know, work? I mean, you had any of the genes pointed towards the immune system, you know, these genetic risks that pushed you one way or another, or was it just a sort of sudden inspiration? You suddenly thought, well, I'm going to go completely different. This is where it's all at. It's all to do with inflammation.
If I remember rightly, Roger, I think you published a paper a long time ago suggesting that Parkinson's might be an autoimmune condition. And that was, I can't remember the date of that, but that was
1988. It was actually when I was a junior doctor, having done no research at all.
Yeah. So this theory was sort of around a little bit, but it hadn't really gained much traction at that stage when I started looking at it back sort of around 2012, 2013. But we had done this a small genetic study where we looked at all of the patients who'd been through our research clinic over the years. So we had a group of, I think it was about 500 actually at that stage, who'd had DNA collected. And we looked at whether there was any link between variations in a particular genetic region called the HLA region and risk of getting Parkinson's disease. So HLA, this stands for human leukocyte antigen. It's a region which encodes a protein that's really important in terms of helping the immune system to recognize specific sort of damage signals or disease signals.
And that's the area that's often associated with diseases like rheumatoid arthritis and you know, autoimmune diseases, autoimmune thyroid disease and things like that.
Absolutely. Yeah, it's linked to a whole host of these different immune diseases, and also it's been implicated in multiple sclerosis.
Right. And
actually we were working with we had some colleagues at the time who were interested in MS, and they've been looking at these variation in HLA in their MS cohorts. And we decided to sort of work with them and explore that in our Parkinson's cohort, because there had been these sort of suggestions in the literature, one of them being from you yourself, that, you know, the immune system might have something to do with PD. So what we found was that actually there did seem to be quite a strong link between variation in this particular region of the genome and getting Parkinson's disease. And around the same time, another group in New York actually found exactly the same thing. They published that at the same time as we did back in 2010. And since then, that's been replicated in quite a lot of different studies. They've all consistently shown that this HLA region is linked to PD, and a whole host of other immune genes have now been linked to Parkinson's as well. So it's quite strong evidence that actually, you know, if you have something different about your immune system, it alters your risk of getting this condition.
Okay, so your sort of general hypothesis, and we'll come back to it specifically in a minute, is that the immune system isn't just there to mop up the damage that happens in Parkinson's. The immune system, your response, if you like, how your immune system reacts to disease, is important in how it actually drives the condition. It's actually important in explaining possibly why you get it and then why you follow different courses. And that's been your main area of work, and we'll come on to the projects which link to that in a minute. But if it's all right, I'm going to ask Nina next a little bit about herself. So there's a very international group we have here, I have to say. So Nina, tell us a little bit about yourself, how you came to Cambridge and how you came to work with Caroline on this.
Right, so I'm originally from Greece, so I grew up in Athens. And that's where I did my undergraduate degree. So I studied biology at first. And then I decided to move to the UK because my main interest from my undergraduate study was neuroscience. So I went on to London and I did a master's there in neuroscience. And right after that, I actually joined you, Roger, and Caroline. So I started my PhD here in Cambridge with both of you as my supervisors at that point. And the reason that I decided to pursue this study was I was really interested in Parkinson's disease, also from personal reasons, because a close family member of mine suffered from Parkinson's. But also I found it very interesting how we call it one disease, but in fact, it's so variable, as Caroline was just saying. We see people having very different symptoms. In some people, the disease kind of progresses quite slowly, and they do quite well for a long time, whereas others, it gets more aggressive, and they also develop memory or thinking problems. So this idea kind of formed the core of my PhD studies.
So you were interested in neuroscience, then you had someone who's with Parkinson's, and you thought, I can bring these two together.
Yes.
And then picking up on this theme that Caroline was saying, that, why do people follow different courses? And so in your PhD, you pursued that in what sort of way? And we'll come on to more specifics in a minute, but what was your PhD largely around? Was it around studying patients, studying mice?
So the biggest part of my PhD had to do with tissue from people who had passed away with Parkinson's disease. So we are quite privileged here that we have a tissue bank that provides us this brain from people who had the disease. So then we can actually study what's going on in the human brain, because a lot of times, a lot of the models that we have, the animal models are helpful, but they don't completely replicate the disease. So our best chance is to actually look what's happening in the brain.
I'm going to stop you there, because I want to keep people on the edge of their seat as to what did you find in the brain that was so exciting that will help Caroline answer her question. I'm now going to move down to Julia. So Julia, tell us a little bit about yourself. So how did you end up in Cambridge? And how have you ended up getting involved with this trial, which we're going to hear a little bit more about later on, but how did you end up coming here?
So I suppose a little like Caroline, I have two hats, and so I was a trainee doctor. I trained in Newcastle, graduated in 2011, but have always
That's
been
right up the north of England, isn't it? That's
oh yes,
way.
Yes,
yeah.
Sorry, yeah. 300 miles north of here.
Yeah.
So I've always, I suppose, since a medical student, I've always been interested in neurology and wanted to be a clinical neurologist. And I ended up moving to the southeast of England and knew that there's a great training program to be a clinical neurologist based in Cambridge, which incorporates research or as part of your training. And I'd done a small amount of research in the group in Newcastle, looking at Parkinson's, and I'd really loved it, found it very interesting, and essentially wanted to be involved in Parkinson's research, and was lucky enough to get a job a training job in Cambridge to train as a clinical neurologist, and got in touch with, you, Roger, and Caroline, to find out about research opportunities, and joined the group in 2018. So I came on board as a PhD student, taking time out of my clinical training to do a PhD, and essentially my role was to try and get a clinical trial up and running in the group,
which has proven to be quite exciting in the COVID era.
Yeah, it's been a bumpy road, but well, as we'll talk about, we're just about there.
Yeah.
There's been a few hurdles on the path, but no, we've.
And can I just ask you about your time in Newcastle? Because Caroline and I have a sort of long-standing association with the team in Newcastle, so we do a lot of work around Parkinson's together. Mm-hmm. And so your work there was on Parkinson's disease itself, or was it a sort of broader topic?
No, so it's on Parkinson's itself. So I worked in the group with David Byrne on one of the cohort studies, which I think was running in Cambridge as well at the time, the ICL study. And I had a small sub-project looking at cognition and gait and walking in Parkinson's, and got to play around with great sensors in a gait lab, which was wonderful fun.
Gait as in walking, as opposed to something you hang in your garden.
Yeah. Absolutely. Good eye. Yeah.
Great. Okay. Well, we'll come on to your trial in a minute and some of the trials and tribulations of that. Marta. Now, Marta, always nice to see you. So tell us a little about yourself, because I remember you coming to us from Portugal. So But tell us a little bit about your background and why you decided to leave the beautiful sun and warmth of Southern Europe to the damp, cold rain of the United Kingdom.
Most days I do not regret that decision. So I studied clinical psychology in Coimbra University, which is a very old academic town in many ways similar to Cambridge. And then I trained as a neuropsychologist and worked as a neuropsychologist in several places. The longest time would have been in Lisbon at the Champalimau Foundation, which is a big neuroscience institute. And they had a private clinic there. So in the mornings, I did clinical neuropsychology and I assisted patients in assessing their memory problems and also trying to rehabilitate them. And then in the afternoon, I went up one floor and collaborated in their research neuroscience program. And I did I focused mainly on food reward behavior in 3 sample groups: severely obese, people with obsessive compulsive disorder, and Parkinson's disease, all of which dopamine is a key neurotransmitter. And I was, excuse me for the false modesty, but I was being very successful in my private practice at the neuropsychology unit, and I was offered a full-time position. And to which point I had to decide whether I could give up research. And so instead of accepting a very nice for life job, I said, actually, I'm going to turn it down and I'm going to pursue what I had in the back of my mind for a long time, which is a PhD. And so with their support, I stayed as long, working there as long as I found the wonderful opportunity that I did was to work for you and Caroline here in Cambridge. So I moved to Cambridge in 2017, and I am working on the role of the gut and constipation and gut health in Parkinson's.
So you've moved to the other end now.
I have. Exactly right. Yes.
Yeah, but the gut obviously is something very dear to you. So we'll come back to that in a minute. So great. So people have arrived from different places, through different routes, through different opportunities and personal sort of exposure, if you like. So coming back to this sort of central idea, Caroline, about the immune system in Parkinson's. So, you know, this, as you say, has been around for years. And a lot of people have said, you know, whenever you damage something, say you cut yourself or something, obviously the body has to heal. And part of that healing process is the immune system comes along, removes the debris, allows things to recover. Now, obviously, with brain diseases, you don't recover, but there's always been this feeling that inflammation just follows the natural death of these, or the death of these cells in the disease. So what you're saying, I think, if I've understood it correctly, and it'd be useful to hear your exact theory, is that inflammation is not just a bystander coming along clearing up the rubbish, it's actually a central driver of the disease. And if that's the case, I suppose the question is, well, what's driving what? What makes your immune system respond, and what is it responding to? Because normally, if I get COVID, heaven forbid, then it will be the COVID that causes my immune system to react. It's the bug that causes it. So what's driving these problems in Parkinson's, and I suppose, how does that explain some of the differences, do you think? And then we can come on perhaps to discuss how you're exploring that idea.
Yeah. Well, I think the honest answer is at the moment, nobody's entirely sure what's driving the immune response in Parkinson's, but it's very clear that it's there. So we know that it's there because if we look at the brain at post-mortem, I won't say much more about this because Nina will, but if we look at the brain at post-mortem, we can see it's inflamed. We can see if we look at the brain with PET imaging using a special ligand that binds to microglial cells or cells of the immune system in the brain, we can see that they are activated. So we know that there is inflammation there. We also know that there's inflammation peripherally in the blood. So inflammatory markers are raised in the blood, immune cells in the blood in Parkinson's look activated. And so we know all this is going on, but as you rightly say, the critical question has been, you know, is this just a sort of bystander, you know, secondary effect of the fact that you've got a brain that is undergoing degeneration? Or is it actually more important than that? And is it sort of driving the disease process?
So just to pick up on that, so you've got inflammation in the periphery. So it's not just inflammation in the brain, where you've actually got inflammation which you can pick up in the blood, that there are changes in inflammation in the blood. So it would
Yeah.
imply it's a sort of systemic problem, like you would if you had any infection, if you like.
Yeah, absolutely. And that's one of the sort of critical bits, I guess, of our hypothesis, is that it's not just inflammation in the brain in Parkinson's disease that's important. It's the fact that the immune system's activated in the periphery, and that peripheral immune activation in the blood is sending messages to the brain to increase inflammation in the brain itself. Also, that those immune cells in the periphery might be causing direct toxic effects by getting through the blood-brain barrier and interacting with neurons, with brain cells themselves. So we think this peripheral immune activation is playing a really critical role in terms of driving pathology in the brain and then driving how fast the disease progresses. And there's various sort of lines of evidence that we'll probably come to when we talk about individual projects that help support that idea that peripheral immune activation is driving this sort of central process of pathology and what have you, and disease progression. But going back to your question of then what's driving the immune response in the first place, what's driving that peripheral immune response? So there's accumulating evidence that alpha-synuclein is important in that. So alpha-synuclein is of course the protein that plays a critical role in Parkinson's disease. We know that it aggregates in the brain in people with Parkinson's disease. But you can also find aggregates of this protein in the periphery. So we know that you see aggregates of alpha-synuclein in the gut, and Marta will probably come back and tell us more about that later. But we can find these aggregates as well in the blood. So we've been doing some work here with our colleagues in the chemistry department, looking at very, very tiny aggregates of protein in the blood, and we can pick these aggregates up, and they seem to be present more in people with Parkinson's than in people without Parkinson's. These aggregates also look different. They're larger in people with Parkinson's versus those without. And then various experiments sort of in the lab show us that if we put these aggregates with cells, with immune cells from people with Parkinson's disease, we can see that these immune cells get activated. So we know that alpha-synuclein can activate immune cells in a dish. And it seems to do that a little bit more in people in immune cells from people with Parkinson's disease.
So these are aggregates you wouldn't normally, because alpha-synuclein is a common feature of everyone's brain.
Yeah.
But this is undergone some change. It started to aggregate, and is it sort of leaking out of the brain into the circulation, or is it being formed in other parts of the body and getting into the circulation through other places, or so don't we know?
I mean, probably a bit of both. So in studies that have been done in animals, this is not our own work, but in the literature that there's evidence out there to suggest that alpha-synuclein can leak out from the brain into the lymph nodes, the cervical lymph nodes, or the lymph nodes in the neck, so it can get out into the periphery. But also, as I said, we know that alpha-synuclein aggregates in the gut as well. So there's plenty of alpha-synuclein in an abnormal form out there, outside the brain, for the immune system to see and for the immune system to be activated by. And there's been quite a bit of work done now to suggest that there is definitely a specific immune response to alpha-synuclein that can occur in normal people, but occurs to a greater extent in people with Parkinson's. So our own work that Kirsten Scott, who was a PhD student here with us, was doing was looking at antibodies to alpha-synuclein, and she showed that there are increased levels of alpha-synuclein antibodies in Parkinson's disease. And then work that David Saltzer's group have been doing in the States has found that there is a specific T cell response to alpha-synuclein as well in people with Parkinson's disease. So we know that. Sorry.
But I was going to say, I would have thought all that was good news, because normally if you mount an immune response, if I mount an immune response to COVID, it might not be the best example because that normally does you more harm than good. But normally if you mount an immune response, you kill things off. So you would say, actually, you're responding to this alpha-synuclein is killing it off, and therefore it should be stopping you getting Parkinson's or stopping you progressing.
Yeah, well, that's a really interesting question, isn't it? Whether that immune response is having some beneficial effect or whether the effect is entirely detrimental. I mean, part of the reason that we think it's detrimental is that if we look at immune markers in the blood, so we did this in a group of people with Parkinson's collected in Cambridge and also in Newcastle. So this is part of the study that Julie was just talking about in Newcastle, called ICICLE. So we've got about 250-ish people involved in this study. We've collected blood samples from these individuals at the time of diagnosis, looked at their profile of immune markers, and we see that individuals who have a sort of inflammatory profile, they seem to progress more quickly. So if we look at their disease trajectories over the next 3 years, we can see that the people who've got raised inflammatory markers in their blood at diagnosis do worse in terms of their progression of motor symptoms and their memory and thinking problems. So that's quite a powerful bit of evidence to suggest that actually immune activation is bad news, certainly early on in Parkinson's disease. But we haven't yet done that in terms of looking at sort of specific T cells for alpha-synuclein and whether they are good or bad. We still don't know
specific
that.
parts of the immune system. So I'm going to move on in a minute to Nina. So the theory would be that this alpha-synuclein aggregates, wherever they come from, brain, in the periphery, the gut, they trigger an immune response. And in certain people, that immune response is actually detrimental. It makes things progress more quickly. And so if you mount such a response, your disease will progress at a rate that's faster than other people. That's the hypothesis which you're now investigating. And one of the questions, and do put your questions in the chat, is about, someone wants to know about how they can find out about autoimmune disease, which obviously you can do through having blood tests to look for specific antibodies against specific things like the thyroid and such like. But in Parkinson's disease, is there any evidence that autoimmune disease is more common? So, if people, for example, have asthma or eczema, they can often have, it's more associated with rheumatoid arthritis, diabetes, thyroid disease. All of these autoimmune diseases tend to go together. Is there any evidence that autoimmune disease is associated with Parkinson's or not?
Sorry, Roger, was that a question for me or for Nina?
It was a question for you, Caroline. Sorry.
Oh, sorry.
Yeah.
Sorry. So the answer to that is it's not hugely clear that there's a link between autoimmune conditions, specifically in Parkinson's. But what does seem to be the case is that if you would take drugs to suppress your immune system, that's associated with a reduced risk of Parkinson's disease. Okay,
so it's looking at slightly.
Yeah, so there's been a couple of fairly convincing epidemiological studies looking at many thousands of people who are on immunosuppressants, showing that their risk of then going on and getting Parkinson's disease is lower. But those kind of studies are quite tricky to interpret because, we don't really know whether it's the immune drug itself that's causing that reduced risk or whether it's the fact that you're taking an immune drug for an autoimmune condition. So where is the link? And it is quite tricky to sort of decipher all of that.
And we might come back to that because often one of the questions which arises naturally from this discussion is from people with Parkinson's: should I start taking, ibuprofen? Should I start taking, anti-inflammatory drugs? Because if you're not sure, it's probably better I take it than not take it. But of course, these are not without side effects. So Nina, I mean, you came to Cambridge to do this PhD looking at the brains of people who had passed away with Parkinson's disease. And so normally I always think of the brain as being pretty quiet on the immune front. I mean, luckily it's protected from the immune system, not much goes on. But, based on what Caroline's saying, you'd expect there to be quite a bit of activity in there when you look at it under the microscope, that there must be immune activation. So what did you find and what did that lead you to sort of think?
Yeah, so there's this idea that the brain is sort of an immune privileged area, but a lot of recent studies have shown that's not quite true, and that there is a lot of immune system components in play in the brain. So what we did in this study is that we collected a lot of brains from people who used to be in the clinic, so people who had been seen from you and Caroline while they were alive, so that we had a lot of clinical data as well. We knew how the disease progressed in all of those people. And what we wanted to see is if we divide those people in sense of their symptoms, people who had a more aggressive Parkinson's with dementia as well, so people who had developed memory and thinking problems during life, compared to people who had Parkinson's but did not develop these memory and thinking problems. What is the difference between these two groups of patients if we look into their brains? And so we did quite an extensive exploration of different immune components in the brain. So we looked at the resident immune cells in the brain, which are called microglia. And we counted these cells in various different areas of the brain. So we counted them in the substantia nigra, which is the main area of the brain where brain cells are dying. But we also looked further out where people hadn't looked before. So we looked in other areas like the outer kind of side of the brain, which has to do with more thinking and memory processes. And what we found is that there was an increased activation of these immune cells in people who had dementia and in Parkinson's compared to people who had Parkinson's without dementia. And we also found that these immune cells seem to be producing more inflammatory mediators, what we call inflammatory cytokines. And at the same time, what we found that was really interesting is that some cells of the immune system, which are normally only in the periphery, so these T lymphocytes that Caroline was talking about earlier, seem to be migrating from the periphery, from the blood into the brain. And that was more severe in the people who had dementia as well.
And if you look at people who didn't have Parkinson's at all, would you see any of that? Do you normally see, as you get a bit older, do you normally see these cells in the brain? Do the microglia normally get a bit out? You wouldn't expect to see any of that.
You wouldn't expect to see any, maybe just one or two odd cells here and there, but it will be very, very sparse. And in most brains of people without Parkinson's, you wouldn't see any.
So when you were looking at these post-mortem brains, there was inflammation, both resident, if you like, cells in the brain, which are normally there to perform immune function, become more activated, and things from the periphery had come in. And that was increased in Parkinson's, but it was even more florid in people who'd gone on to get Parkinson's with the dementia. Yes. Okay. And so, and that was that everywhere, did you say? Because you obviously looked at different brain regions, or was it localized to certain areas more than others?
Yeah, so it was in certain areas more than others, as you say. So it was, we found that, as I said, in the substantia nigra, which is the main area of interest, but we also found it in the amygdala, which is an area of the brain that's kind of associated with emotion, fear, and some aspects of cognition as well. And we also found them in the cortex of the brain, so the prefrontal cortex that has to do with kind of problem solving, this type of kind of higher processing.
Okay, and so that's all found in post-mortem brain, and obviously that's very helpful, but that's like a sort of snapshot. And how do you know that's relevant to what you see in life, if you like? So Caroline's already said you can go looking in the blood and you can pull out markers of inflammation. So I can go looking for the cells that you find from the circulation in the brain in people. But how do I look for these resident microglia, which you're talking about, these resident immune cells? I think Caroline mentioned something about PET imaging, so sort of brain imaging. I mean, have you got involved with that? And what, if so, what are you looking at there?
Yeah, so after I finished my PhD, I stayed on with a group as a postdoc, and now my main project has to do with patients who are newly diagnosed. And what we do is we try to find these immune cells, these microglia in their brains with non-invasive methods, which is essentially this PET brain imaging. So how we do this, we inject a small tracer, which then travels to the brain, and then whatever you see these immune cells, they will give us a signal. And this is what we're doing. So we are recruiting newly diagnosed people, and we kind of stratify them into two groups, in people who we predict they will have a more slow disease progression, and another group of people who we think will probably have a more aggressive disease and potentially may develop memory problems as well.
And that's based on clinical criteria. So when you look at the patients, there are certain things which they cannot or can do in certain ways which predict where they're going to go. Is that right?
Exactly. So this is based on a study that Caroline did a few years back, where this campaign study that she was mentioning earlier, where she looked at a big group of people and followed them up for about 10 years. And what she found is that people who perform badly in certain memory tasks and certain cognition tasks at baseline, so when they first come to the clinic, and people who also have a specific genotype, and that is a specific gene called tau. People who have that genotype tend to do worse later on. So we use those markers to stratify our patients in this study that I'm working on.
So you're saying people are going to do badly or going to do well based on clinical criteria, so how well they do certain tasks and certain genetic variants, but you're not stratifying them into these groups based on inflammation, but you're looking at inflammation in those two groups.
Yes, exactly.
And then I suppose my final sort of question for you at the moment, Nina, is how do they relate to each other? So do changes and you've obviously, you probably don't know the answer to this because you've only just started the study, that to what extent does inflammation in the brain, so with your PET imaging, looking at these brain scans and microglia, match what you see in the periphery? So if I have lots of inflammation in the periphery, do I have lots of microglia activation on my PET imaging, or do these tend to run slightly independently of each other?
Yeah, this is a very good question, and I think we will be able to answer that as well, because at the side of the PET imaging, we're also collecting blood and spinal fluid from all of our patients. And what we're going to do at the end is kind of compare and do see if there is associations between markers in the blood and markers in the spinal fluid compared to what we see with the PET imaging in the brain. And we'll be able to answer that question.
Okay, so all of those studies are going to tell you whether there is a problem. I guess the question is, it won't tell you whether that's a critical problem, because all you'll say is I'm describing what's going on in different types of patients, which brings me to Julia, I suppose, because if you ever want to show that something is causally related, so in other words, if increased inflammation is causing bad things in Parkinson's disease, then there's two ways I suppose I can prove that. One is I can speed up inflammation, which I think the ethics board would probably have some questions about, giving people deliberately infections to see if their disease goes faster, or actually the more sensible approach, which would be to stop inflammation, as Caroline was already saying, by using drugs which suppress the immune system and seeing what effect that has. So Julia, how are you tackling this? So what sort of, when you talk about, you know, stopping the immune system, is this just an aspirin every other day? Is it a bit of steroid? I mean, how are you approaching it, and what sort of drugs are you using, and how are you seeing whether it has an effect, or can you do that at this stage of sort of looking at this particular problem?
So the clinical trial that we're running that's just opened is using a drug called azathioprine. So this is a drug which targets the immune system and suppresses it in a fairly broad way. And this builds on what Caroline was talking about in terms of.
So this is a special drug. This isn't something I can buy over the counter or go and get from my local chemist.
No, unfortunately, it's not because it does have significant effects on the immune system. So we are using it because we have good knowledge that we think it's going to help, but we do need to monitor it carefully, and that's part of the trial. Is built into our trial to make sure that our patients are safe on the drug and that we're monitoring the response.
And is this a new drug? Is this something which is, just arrived on the clinical scene, or is this something that's been around?
No, right, it's been around for a while. I think in the 1960s.
Right, even before me.
So it's actually widely in use in clinical practice for a whole range of conditions. So we know a lot about it. We know about how to monitor it and what it does to the immune system. We use it in conditions like rheumatoid arthritis, like inflammatory bowel disease, like psoriasis. But we haven't used it in Parkinson's before. But all that wealth of knowledge means that we know quite well how it works and how to monitor it, which helps with running the trial. So
this is a well-established, this is a drug which has been around for a long time, that sort of has a major hit on the immune system. So it's not like a sort of non-steroidal, if you like, or a bit of aspirin or a bit of ibuprofen. This is a big player that gets into your bone marrow and different places and stops cells being produced. So it has a fairly major effect on the immune system, but it's relatively safe in the sense that we know what to look for. And so is your study just saying, if I give it to people with Parkinson's, hopefully they'll survive with it, and that's all I'm interested in? It's not too toxic for them, or how are you going to know whether it's having an effect on the immune system in people with Parkinson's and that's having an effect on the disease, or is that something you can't really answer in this trial?
Well, we do hope to answer that to a degree. So what we're doing this trial, it's I suppose what called a double-blind trial. So it means that there's a group who are on the medication and there's a group who aren't on the medication who get what's called a placebo. And neither me or anyone who's running the trial or the participants know which drug they're taking. And what we think is that the people who are on the azathioprine will have the disease slowed down or stopped. So at the end of having when we come to analyze the trial, we'll be able to look at all those people who've had the drug compared to those who didn't, and see where they are in terms of their disease and compare them.
And how will you do that? Is that, do you just ask them, or do you sort of measure their time they take to walk somewhere? I mean, how will you assess that?
Well, I suppose, as we all know, the major way that Parkinson's affects people is to do with their movements. We use lots of rating scales, which are currently really commonly used in other clinical trials. And I will rate them all the way through the trial in terms of the primary endpoint is looking at movements and is looking at movement and slowness. But there are lots of other aspects of Parkinson's that we're capturing as well, because we know that it's not just a motor condition. We are looking at the non-motor symptoms, memory and thinking. And the other thing we're doing is looking at the immune system and what impact we've had by giving people azathioprine on those changes that we've that our group and other groups around the world have found on the immune system, both by looking at blood cells and inflammatory markers in the blood, but also looking at spinal fluid and PET imaging. So we'll hopefully have a wealth of information to look at and see the effect of this drug.
So you're taking all the, so, you know, as Caroline was saying, you found these changes in the blood. I mean, the drug obviously works systemically, so you would expect something there, but you're monitoring that. And then obviously Nina's doing this imaging of the brain to look at these microglia, these resident cells, and you'll also be looking at that in your patient group. And as you say, nobody knows what anybody's having. So I suppose one question that arises is, you know, that sounds great, but isn't it a little bit couldn't it be a little bit hazardous, because you don't really know what's going on, and some of these drugs are, you know, azathioprine's quite toxic. So how do you get around that problem?
So we have got around that problem by having a team of doctors who are unblinded, who know what treatment people are on, and they're looking at the results, making sure that everything is on track. And of course, if the worst happens, and for whatever reason we need to find out what treatment our patients are taking, we have a system that can be accessed 24 hours a day to unblind that person in an emergency. So there's lots of fail safes to make sure that everyone is safe on the treatment.
And the idea would be, I guess, that if this is, and this relates to a question I've received, you know, if it works to slow down the disease, it wouldn't just be slowing down one aspect of it, it would slow down all aspects of it. So whether it's movement, thinking problems, all of those, you would expect them all to slow down.
I mean, we would really hope so.
Yeah, and all of that's been captured in your trial.
It is, yes. So we've got quite a wide range of clinical outcomes that we're looking at. We've made our primary outcome, the motor score, which previous research has shown is the most sensitive or the best way to pick up change in Parkinson's. Because we do acknowledge it's an early trial, it's just running for a year, and we want to be sure that we can that we're not going to miss the fact that this drug is making a difference. Okay. We want to pick up a really sensitive measure to make sure that we're picking up everything.
And if that works, is that the end of the story? Can we stick everyone on azathioprine and thank you very much, we're all done?
Well, I suppose I'm mostly familiar with how things work in the UK. It would need to be approved by say if it did work, it would need to be approved by something like NICE. It's likely they're going to need a bigger trial, or a phase 3 trial of many more participants. We're at the moment recruiting 60. Likely that we'd need more evidence before we can, give everyone azathioprine. Yeah.
So I guess your trial is a sort of first step, that if you got all the positive signals, you'd say this has mileage. Let's do a proper study now with hundreds of patients and show that it really could slow down disease. And if so, then we would be able to recommend it for certain people with Parkinson's. So that's great. So Martin, how do you fit into all this? So we've been hearing we can measure inflammation in the blood, we can look at inflammation in the brain. Nina's looked in the brain and shown inflammation. Julia's going to intercept this inflammation it was sort of tantalizingly mentioned by Caroline that Parkinsons may not begin in the brain, or at least the alpha-synuclein aggregates are found elsewhere. So tell us about what you're doing in the gut. Why should I be interested in the gut? And for people who are really interested in this, as I say, on the 1st of September, we will be discussing this even more with a team from Denmark. So Marta, how does your project fit into this, and what are you thinking might be driving inflammation on the immune system in Parkinson's disease?
Well, I think a very likely culprit of this low chronic inflammation in Parkinson's disease could be the gut. In 1817, just a couple of years before you were born, Dr. James Parkinson, on shaking palsy,
my dear brother,
yes, suggested, actually reported that he prescribed laxatives to his patients with shaking palsy, and their complaints were temporarily removed. And he actually wrote that he wondered whether the gut and the brain were connected. So 200 years later, we're still debating the issue, but we have more evidence to suggest that it is. So a lot of participants, and I should say a lot of people with Parkinson's disease have constipation even before the motor onset of the disease. There have been case reports showing alpha-synuclein in the gut of people who without Parkinson's disease that 20 years later will develop. So these were colonics, so biopsies from the gut, they were stored and frozen for other medical reasons. And then when the patients developed Parkinson's disease, they went back and they saw alpha-synuclein then. And we know that alpha-synuclein is associated with inflammation in the gut. Constipation is also associated with inflammation in the gut. And in Parkinson's disease, the disease has been associated with what we call the leaky gut. So the walls of the gut are permeable. We have trillions of good and bad, but of bacteria that live within us, and they produce our vitamins, they digest our fibers. They're really, really healthy for us. But in a leaky gut, so the walls of the gut, the wall is slightly more open. So all of these good bacteria that we don't recognize as foreigners leave the gut and go into the bloodstream. Then perhaps the immune system, what it does is it raises up a little bit the immune system as into alert and just clear out the debris, clear out the bacteria that are good, but are just not in the right place. And that potentially might explain what Caroline saw in the icicle study, where in the absence of any infection, the participants were not sick, but their immune system was raised. So
do you think, so the immune system is raised, do you think, to the leaky gut and the bacteria and such like coming out of the gut, or aspects of bacteria, or do you think also the synuclein is leaking out of the gut? Because you said that people have synuclein in the gut.
So those are not mutually exclusive. So
Right.
I don't know the answer to this, but could be one, could be both, could be multiple systems playing up together. Another thing that was mentioned before is genetic susceptibilities. So people with constipation and people with Crohn's disease are more likely to develop Parkinson's in the future. And a specific gene called LARC2 has been both associated with Crohn's and with Parkinson's.
So Crohn's is quite interesting. This because Crohn's disease is an autoimmune disease.
And azathioprine is recommended for Crohn's.
Yeah, and you give people immunosuppressive, but actually Crohn's disease itself is linked to a later risk of getting Parkinson's disease.
So we're coming full circle now within the group.
And do other disease, so Crohn's disease is similar to a thing called ulcerative colitis, which is another inflammatory bowel disease. So do people with ulcerative colitis also have an increased risk of disease, or is it only Crohn's?
The studies mostly point towards Crohn's. Okay. There have been some studies looking at irritable bowel syndrome and colitis, but I don't think the evidence is so strong.
Okay, so your theory would be that so bringing it all together, if we wanted to have a unifying theory, you would say years before, and I think you've just recently had a paper on this, that you know, if you look at levels of constipation at onset of disease, that's a predictor of disease course, i.e. the more constipated you are, you tend to do less well. Caroline says from her studies, the more inflammation you have in the periphery, the worse you tend to do. So those two seem to be linked together. But the idea would be that something goes wrong in your gut early on, that makes it a bit leaky, let's say, alpha-synuclein comes out with some bacteria, your immune system recognizes it, it's then geared up to clear it. You start to develop synuclein problems in your brain. It then reacts to that. It not only gets rid of your alpha-synuclein in the brain, it gets rid of your brain cells as well. And then the whole system is sort of this sort of fiery furnace of inflammation, which is driving the disease on. And then Julia's going to come along and pour water on it with azathioprine to just quiet the whole thing down and let it all. That's the sort of general
working hypothesis.
So one of the questions I've got here been sent in is about, anti-inflammatory diet. So I suppose one of the questions which arises from all of this, which I mean, by any means anyone answer, is how aggressive do you have to be? Which comes back to an earlier question about how targeted do you think this has to be, targeting the immune system. How aggressive do you have to be in terms of the therapies? So, there are obviously diets which are anti-inflammatory diets. There's low-level inflammation agents like a bit of aspirin. At the other end, we now have all of these fancy antibodies that people make against this part of the immune system or that part of the immune system, against this cytokine or that cytokine. So I mean, perhaps I'll start with Caroline. I mean, what's your feeling on that? How aggressive do we have to be? How specific do we have to be, or is this for the future?
I think the honest answer is we just don't know at the moment. And the idea behind the AZA-PD trial was just sort of proof of concept that if we suppress the immune system, and to clarify, azathioprine, it's a proper immunosuppressant. It will make changes to immune cells, but it's not really heavy duty immunosuppression. But it does sort of have a rather blanket effect on all sort of proliferating, dividing immune cells. So the idea is just to see whether if we do that, does it make any difference over 12 months of treatment to the course of the disease? And can we see that it's causing any change in terms of what's happening with inflammation in the brain? So that will give us some idea of whether this whole sort of rationale makes sense, that suppressing the immune system is a good idea. I think if that doesn't work, then our next question will be, so what's wrong here? Why has suppressing the immune system not had an effect? And the answer to that then may be, well, because we need to be much more targeted. And it may be, coming back to what we said earlier, that some aspects of the immune process actually are having a beneficial effect and other aspects of it are having a detrimental effect. And so in parallel to what we're doing with AZA-PD, we've got these other studies running, looking specifically at sort of immune cell changes in blood and in the brain through looking at the CSF, trying to tease apart more precisely what's going on, and then can we target more specific aspects of the immune system. So for example, Nina's also really interested in toll-like receptors. So she's published an article fairly recently about toll-like receptors in Parkinson's disease. So these are the specific receptors that we think alpha-synuclein binds to cause activation of immune cells. And we also know that endotoxin, which comes from gut bacteria, also acts via toll-like receptors.
Okay.
So one of the sort of strands of work that we're doing is looking at toll-like receptors and seeing what happens if we block toll-like receptors in cell cultures in a dish. So what impact does that have? Also, if we look at an animal model and we look at toll-like receptors and then block them with a drug, does that affect the course of Parkinson's disease in an animal model? So that would give us a much more sort of specific target in the immune system. So our next question is going to be, is that better, more effective than this sort of blanket approach? So
that raises a sort of quite a challenge then, doesn't it? That you may, if you just have a blanket suppression of the immune system, you may be getting rid of some of the good parts of it as well as the bad parts. So that, I guess, is a challenge. And I remember at some point you had some very intriguing data from these studies you were talking about earlier, these campaign studies, and I think the iSchool study, where people who entered the study who were taking aspirin or non-steroidal anti-inflammatories, brufen and ibuprofen and things like that for other reasons, seem to be slightly protected, at least in the early stage, from developing major cognitive problems and dementia. So I suppose, coming back to my question, it may ultimately be that actually low level, early on, relatively sort of easy to administer oral drugs may have an effect.
Yeah, that's absolutely right. So non-steroidal anti-inflammatory drugs, things like ibuprofen, we found that people who were taking those sorts of drugs at the time of diagnosis seemed to have a slower progression to dementia in our campaign study. It's difficult to know what to make of that because the numbers are relatively small for this sort of epidemiological study. You really need to look at thousands and thousands of people to be able to be sure about whether, you know, these sorts of drugs that are making a difference. But there have been some studies that suggest that is the case, but also others that suggest that actually it doesn't matter whether you take non-steroidal anti-inflammatories or not. So it's all really a bit unclear. And we wouldn't sort of recommend that people do go out and start taking ibuprofen because of the fact that the risk of it causing gastric irritation, even gastric bleeding. So it's not the sort of thing that you should go and just start taking just in case. You have to discuss it with your doctor and sort of think about, you know, possible side effects and things. So I think it's too early for us to say, yes, we all ought to be taking these sort of, you know, anti-inflammatory pills just in case. I think we need to really question this properly in trials. And one of the reasons for going for azathioprine rather than one of these sort of non-steroidal anti-inflammatories is partly because of the fact that we understand better how azathioprine works. So these non-steroidal anti-inflammatories have all sorts of other effects as well. They're rather complicated in terms of how they work in the brain. So the idea behind azathioprine is quite a pure immunosuppressant drug. We know sort of what it's doing, and so we can answer our question of does suppressing the peripheral immune system actually make a difference?
Okay, so I mean, the sort of take-home message is don't rush down to your local chemist and stock up on all of those tablets until we have more evidence for that. So I mean, we're coming towards the end, so if you do have a last question, do stick it in the chat. But I just want to come back to you, Marta, because these drugs, which are immune suppression and things of that nature, I mean, how much, and they irritate the gut, as Caroline was saying, so how much does that change all of the flora and all of those bacteria that live in your gut? So do these drugs really work by suppressing the peripheral immune system, or do they just change what lives in your gut and that makes you a better person?
So that's a really interesting question, and I don't know the answer to. I don't think it's easy to answer for many reasons, because the opposite also applies. So H. pylori infection, which is a bacteria that infects the stomach and may give you or not symptoms. After a course of antibiotics, some people with Parkinson's disease reported feeling better. And so it was posited that perhaps this bacteria infection was at least worsening PD symptoms. But what happens is most antibiotics also have quite a strong anti-inflammatory effect. So we don't know whether the antibiotics are killing the bad bacteria and giving you better quality of life, or actually it's the anti-inflammatory effect. And the same is true for anti-inflammatory drugs. So it could be the dampening down of the immune system. It could be the alterations of the microbiota. We don't know.
Which is the bugs that live in the gut. So our hour is up. Thank you very much for a fascinating, exploration of the role of the immune system in Parkinson's. Thank you very much for all the hard work you've done on that. Good luck with your trial now that it's up and running in the post-COVID. Thank you all very much for listening now or listening later when this is posted. Thank you all for your questions and for being a part of this. Our next session is on the 1st of September, when we will be discussing where does Parkinson's disease begin. You've already had a little tantalizing conversation about that this afternoon. Is it in the gut, or is it in the brain, or is it in both? Who knows? Anyway, 1st of September, have a lovely summer and we'll see you all then. Thank you. Bye-bye.